there, this break. Welcome to part 3 of Chemistry of Life. We will be talking about proteins today, a word that you've probably heard a lot, right? Anyone work out? Anyone trying to get some muscles? You probably take a lot of protein to make your muscles a little bit bigger. But so, what do proteins do? And there's a long list of what they do because actually proteins just do everything in the cell. Proteins do all the work of the cell. So I have a list here that you guys should take a look at and be fairly comfortable with. Proteins, now you may have noticed I put an exclamation mark behind each of these. I guess I got a little excited. Exclamation marks are a bad habit of mine, but here's the list. Enzymes. Protein catalysts are called enzymes, and most of the work that your body does is chemical work. All right, so most of the proteins in your body are going to be enzymes. You remember what a catalyst is from your chemistry course? A catalyst is any molecule that speeds up a chemical reaction. Proteins can be antibodies. They can protect you from molecules that are going to make you sick. Okay, so protein antibodies, when you say antibody, you're talking about a protein. Motor proteins or contractile proteins are proteins that move molecules within your body, or they could actually cause muscle tissue to contract. When I contract my muscle, it's because the motor proteins in that muscle are actually sliding against each other and causing the muscle to shorten. Okay, proteins can be signaling molecules like hormones. An example of a hormone is insulin. Insulin tells your muscle and your liver that there's a lot of sugar in your blood that needs to be taken up and used. Okay, they can be structural molecules. Okay, we have a skeleton. Every one of your cells has a skeleton too. Our skeleton is made of bones. A cellular skeleton is made of structural proteins. All right. Also, your hair and your nails and your skin, they have proteins in them that are structural. They give your skin their tensile strength, like keratin is an example of a protein that gives structure to your skin. It gives strength to your skin. Or they can be transport molecules. They can move molecules into or out of your cell. They could be little holes in the plasma membrane, or they could be, I guess we could describe them almost as hands that grab something on the outside of the cell and put it on the inside of the cell. So these are functions of proteins. So here's our protein. Here's what you should be able to look at and say, this is a protein. More specifically, this is an amino acid. It's one monomer of protein. So we've seen that most of these molecules are going to make up polymers. If you get a chain of these amino acids, okay, that chain is called a polypeptide. A polypeptide, many amino acids in a sequence. So what does one amino acid look like? It's going to have an amino group, okay, NH2, and it's going to have a carboxyl group, COOH. Okay, there's the central carbon to which these two functional groups are attached to. There's a hydrogen atom that's always going to be attached to it, and then we have something here called R. Okay, R represents a variable part of the amino acid. So you have 20 amino acids that your body uses, and every different amino acid has a different side chain. It has a different R group. That's what makes it different. So you take one amino acid, as we saw with carbohydrates, you can join another monomer of protein, another amino acid to this one. Okay, you see what's highlighted here? 2 hydrogen molecules, 1 oxygen molecule. What kind of reaction are we looking at? A condensation reaction, which will form a covalent bond between our 2 amino acids, and we call this covalent bond in proteins, we call it a peptide bond. So now our amino acids are sharing electrons. They have a covalent bond called a peptide bond. Of course, over here, we formed water, as we always do in these condensation reactions. Do you remember what the covalent bond was called in our carbohydrates? It was called a glycosidic linkage. Do you remember that? Okay, so you should remember that for each of these macromolecules. Carbohydrates form a glycosidic linkage. Proteins form a peptide bond. Now, again, we have the different functional groups here. Well, the functional group on this amino acid is very simple. It's just a hydrogen. But the functional group here is a methyl group, CH3. All right, so these are the variable regions in our amino acid. I told you there are 20. Okay, they can be categorized as being nonpolar or polar. And as we've talked about before, a nonpolar R group is going to be hydrophobic. It's not going to interact with water. And a polar R group is going to be hydrophilic. So there are 20 kinds of those, which we have here. Okay, you don't need to memorize them for this course, but appreciate um, the differences in the R groups, in the side groups. So these are nonpolar, and it should kind of be obvious to you that they're nonpolar because you don't see those electronegative atoms in them. Well, sulfur is electronegative, and so is NH, but they're not really at one end of the R group like they are in the polar side chains here, right? Here we have an OH at one end of the R group, another OH here, S here, OH. So these are the polar ones, and I would expect that you should be able to recognize them if you were to see them on a test without memorizing them. I won't ask you, well, is this one tyrosine or phenylalanine? You don't need to know the names, but have some, be able to guess the sort of function, if you will, or the properties of the side group by looking at it. Okay, electrically charged, okay, this third category here, it means that there's an ion in the side chain, and anything that's electrically charged is going to be hydrophilic. Okay, now we say that proteins have different levels of structure. You might say there's different levels of folding. What we've described so far is the primary structure of a protein, a linear sequence of amino acids from one end to the other. So if you look down here, this is supposed to represent a chain of amino acids. You could call a chain of amino acids a polypeptide. Poly meaning many, peptides. Many amino acids joined by covalent peptide bonds. Glycine, alanine, serine, etc., all joined together in a chain. We just call this the linear sequence of amino acids. So when your body is synthesizing a protein, it just puts them together. Here would be one amino acid. Glycine, just one H. Here's a second amino acid being joined there, alanine, with the methyl side group. They're being joined together by peptide bonds to make a long chain. And there you go. That's your polypeptide. That is just a line of amino acids. We say that's the primary structure of a protein. Now, proteins don't just come in lines, as you might imagine. They fold on each other to make globules, or they can make many different complex shapes. And those complex shapes, the higher structures of a protein, so its secondary structure, its tertiary structure, and its quaternary structure are always going to be based on the primary structure because these side groups are going to interact with one another on occasion and cause the protein to fold. So let me just skip ahead 2 slides here. The secondary structure of a protein is due to hydrogen bonding. And proteins can form 2 kinds of secondary structure. They can form an alpha helix, which we see here, or a beta pleated sheet. Helix just means a coil, like a spring, and beta pleated sheet is folded. All right, and the dashed lines here that we see are hydrogen bonds. Okay, so secondary structure is either an alpha helix or a beta pleated sheet, and it's due to hydrogen bonding. Now keep in mind that hydrogen bonds are weaker than the covalent bonds, so they can be broken. Okay, and we'll talk about how they can be broken towards the end of the lecture. What I'd like to show you here is the importance of primary structure and how the primary structure can actually affect the secondary structure. So here we have red blood cells. Okay, and they're supposed to look like this, these nice, neat little biconcave discs. Now, one of the main proteins in red blood cells is called hemoglobin. Maybe you've heard of it. And if you were to change the 6th amino acid in hemoglobin here to another amino acid, okay, so here we have, oof, I hope I don't mess it. I think this is glutamate. Glutamate. If we change glutamate to valine, okay, that one amino acid, okay, will cause hemoglobin to fold improperly, and instead of getting nice biconcave discs, we get sickle-shaped red blood cells like this. Now, the function of red blood cells is to carry oxygen. When they're deformed like this, they are not able to carry oxygen as efficiently through the cell. So you get a disease called sickle cell anemia where you can't carry oxygen properly. The tertiary structure of a protein is the result of many interactions between the R groups. Okay, or it can be an interaction between the R group and the peptide backbone. Okay, this gives proteins a more three-dimensional shape. So when we have the secondary structure of a protein, let me try and draw in a little area here. Let's say that this here is my alpha helix, and then we have the beta pleated sheet here. It's a crude drawing. Well, imagine interactions between those two types of secondary structure, and you can kind of see that here, right? There's many alpha helices interacting. Here we can see alpha helices interacting with beta pleated sheets. Well, now we have more complex structure, right? We have tertiary structure. We have different folds of the protein interacting to give you a globule, a kind of a blob. And this is going to affect the function of the protein. Okay, here we see the different types of interactions. You already heard of hydrogen bonds. You can get hydrogen bonds between the R groups. Hydrophobic interactions are kind of funny. If you were to look at my hand here, let's say the inside of my hand is hydrophobic and the outside of my hand is hydrophilic. Well, what happens if we put my hand in water? The hydrophobic parts are going to hide themselves from water, and the hydrophilic parts are going to be present on the outside, right? This is what a hydrophobic interaction is. Okay? Covalent bonds can form, where do we have it here? Where are some covalent bonds? We don't see it. There could be covalent bonds between the R groups. Van der Waals interactions, probably learned about it in your chemistry class. And here we have disulfide bridges. Here are ionic R groups. Remember, some of the R groups have ions in them, so those interact to give you an ionic bond. Well, this influences the folding of a protein and therefore its structure. Quaternary structure is when several polypeptide chains interact with each other. So what does that mean? Well, here, if you look here, this is a protein dimer. Do you see how they're kind of separate? Here's one polypeptide with tertiary structure, alpha helices, beta pleated sheets. Here's another one. Okay, if these two molecules interact, well, it makes a new protein, now with quaternary structure. So there's two separate parts interacting together to give you quaternary structure. Here's another molecule. Hemoglobin. Remember where we found that? In your red blood cells. Hemoglobin is what we call a tetramer. It's 4 different polypeptide chains. Here they're denoted by different colors. So hemoglobin is a protein with quaternary structure. It's a very complex structure. Keep this in mind. The folding of a protein is due to the interactions in the side chains. Now, we said that covalent bonds are very strong, so those are not going to be broken without some kind of chemical reaction. When you're digesting proteins, when summer's over, right? Probably had a lot of barbecue. For those of you who eat meat, digesting lots of proteins. Well, you have proteins, you have enzymes in your body that will break the covalent bonds in your meat. However, it's very easy to denature hydrogen bonds, for example, and some of the ionic bonds that you would find in a molecule, right? How do you break these bonds? You can raise the temperature, okay? When you fry an egg, what do you notice? Well, the egg looks all liquidy when it comes out of the shell, but when you begin to fry it, it changes its properties entirely. Why? The heat is breaking the hydrogen bonds in that molecule and giving it a different nature altogether. So we say that a protein that is unfolded due to high temperature or high pH is denatured. Here we have a protein, ribonuclease. You don't need to know what it does. But if you look at the hydrogen bonds here, what happens when we heat it up, for example, or we could put it in high pH? Well, we break the hydrogen bonds and the entire protein unfolds. So you get these unfolded ribbons here. So that is called a denatured protein. Remember that word. You can denature other molecules as well. We'll see that in a little while. So remember that. Look over your notes with Buddy. And next lecture we will be looking at lipids.